Agnieszka Czerwonka. And the title of her talk is Immunostimulation of the Model Spider Species, Parasteatoda tepidariorum. So Agnieszka, do you want to share your screen?
Yes, of course, I'm doing that. I hope so. Okay, is everything visible? And okay, thank you. All right, so my name is Agnieszka Czerwonka. I am from the University of Silesia in Katowice in Poland. I am a member of the team of the environmental stress biology in our university. And today I'd like to present you our newest project, which is immunostimulation of the model spider species Parasteatoda tepidariorum. All right, so that species is maybe better known as a house common spider from family Theridiidae. This is synanthropic, cosmopolitan species with sequenced genome, what is very important. And I think it's interesting that our team has got culture of that spider since several years. So we have got old time material for research, and we kept it in still known environmental conditions. And at the beginning, the few information about immune system of spiders. So it's innate, it's non-specific, and it's based on hemocytes reaction. Hemocytes are the cells from hemolymph, and their reactions, their immune reactions are, for example, phagocytosis of microbes, coagulation of wounds, or encapsulation of some foreign bodies. And the most precise tool to cope with microorganisms are the antimicrobial proteins. Okay, so since there is not much information about immune system of spiders, we just made some questions to answer. So the first question is, what is the immune response of Parasteatoda tepidariorum to injury, to infections with bacteria, to foreign body in their abdomen? And second question is, do immune responses differ between the selected developmental stages of spiders? And the third question is, what immune system proteins might be present in hemolymph of our spider? So to deal with those questions, we tested selected experimental groups, the age groups, and the groups with different levels of immunostimulation. The age groups were embryos, juveniles, subadults, and adults. And the levels of immunostimulation were, of course, control groups. So there were non-stimulated individuals, and then stimulated groups, spiders with sterile nylon fiber in their abdomen, and the last one, stimulated plus bacteria group. So there were spiders with nylon fiber, which were coated with Micrococcus luteus, again stacked in their abdomen. and the stimulated groups were kept with nylon fiber in abdomen for 2 hours. The material of our tests were hemolymph of tested spiders, and we had 3 methods. So first one was a PCR reaction, in which we tested, we checked if there is expression of genes encoding defensins, lysozyme, autophagy-related protein, and denamine proteins. Then the next one was ELISA test. It's enzyme-linked immunosorbent assay. And we checked, we detect the proteins. The first one was lysozyme, the second one was HSP70, which is heat shock protein. It reacts to physiological stress in organism. And the third
Agnieszka, you got muted.
Oh no! Oh no,
okay.
no!
But it was only for a few seconds. You're okay.
Okay, okay. Now I am. It's okay. All right. Dobra. So where I stopped my voice.
Well, that's okay. Just start this
Okay,
slide again.
continue. Okay. All right. So the 3 methods we used was PCR. Did I said that? Oh no. No. Okay, again, PCR. We checked expression of genes, encoding few proteins of immune system of spiders, which are lysozyme, defensins, autophagy-related proteins, denamine. Then we detect proteins lysozyme and HSP70 protein, which is a heat shock protein. It protects cells from the physiological stress. We detect it by ELISA test, which is enzyme-linked immunosorbent assay. And the last method we used was HEMOCYTOMETER. So the Neubauer chamber, which we use to just count the cells of hemolymph and check their viability. Next slide. Okay, results of PCR. As we can see, we detect lysozyme 1, lysozyme M1, and autophagy-related peptide in all age groups of Parasteatoda tepidariorum. There were also defensins, especially defensin 2, but it's not well visible at this picture. And lysozyme C was only detected in adult spiders. So now we know that spiders, that Parasteatoda tepidariorum spiders can translate proteins to lysozymes, to autophagy-related peptides. So they have got tools to deal with microorganisms. And results from ELISA tests, that test is both quantitative and qualitative. So we know that lysozyme is produced in the highest level in stimulated and bacteria infected spiders. And it's both in adult and juveniles Parasteatoda TEPIDARIORUM spiders. And also HSP70 protein is in the highest level in spiders treated with bacteria and the foreign body in their abdomen. Okay, results from HEMOCYTOMETER was like that. So to check if there were death cells, I used trypan blue solution. And if there were dead cells, they absorbed the stain of trypan blue. And then we know how many live cells and dead cells were in our experimental groups. So the control group has got some dead cells, but with the levels of stimulation, also the level of the amount of dead cells increased, and in stimulated plus bacteria cells of hemolymph, we have got the highest level of dead cells. Those are very first tests in that project, so it's not completed. We are processing those tests, but it's very promising, and we can conclude that, of course, adults and juveniles of Parasteatoda TEPIDARIORUM have got the potential to produce antibacterial proteins, and the bacterial infection increases the level of lysozyme and HSP70 protein, so it causes stress in our spiders. It's obvious, but we have got proof to that. And we also know that immune system reactions to injury and infections are very rapid, and coagulation of the hemolymph and melanization of hemolymph was really quick, and we can see that on that photo that I just made. And what is the last conclusion? That the immunostimulated spiders, only 2 hours after infection with Micrococcus luteus, have got really increased hemocytes, increased amount of dead hemocytes in their hemolymph. Okay, so our further research plans are to test at first other exposure times to immunostimulation, longer exposure times, check reactions of immune system to infection with other species of bacteria, and other concentration of bacteria, of course, and maybe perform the same tests on other spider species, for example, Pardosa lugubris, which we are working with also. And that's it. Thank you very much. Okay.
Great. Thank you, Agnieszka. We have just a little over a minute left, and there's a few questions.
Sorry.
We can take one question.
Okay.
So the first question I saw is, I think it was Jillian Cowles asked, why did you use Micrococcus?
Okay. So the very simple reason is we have got that bacteria, and also, am I muted? No.
No, we
okay.
hear you.
Okay. we have got that bacteria in our lab, and we performed tests in the past in which we saw that this bacteria causes some reaction in hemolymph. So we just have available bacteria in our lab, and we choose it, just that. So it was, like I said, it was pilot studies. So we were just checking, and we need to extend it, of course, to other bacteria species. But it's very common. It's in the ground, it's in the air. So we choose just a very common bacteria.
Okay, great, thanks. To stay on time, we should move on to our next speaker.